You are welcome to today's class. Today we are going to talk about special senses. With a particular reference to, I will talk about taste, which is also known as gustation, the physiology of taste, which is also known as gustation. And then if we have enough time, we will go into the physiology of olfaction. Olfaction. So we'll talk about these two things today. And then in our next class, we are going to talk about vision, which of course will include the visual pathway. We'll talk about the visual pathway. And then we'll talk about the physiology of audition, which will include the auditory pathway. The auditory pathway. Right? But for today, we are going to focus on taste and olfaction. When we are done with this, we'll now proceed to vision and audition in our next class. So let's talk about taste. The physiology of gustation. Now the question is, what is gustation? The word gustation also means taste. So taste is also known as gustation. And gustation means taste. Now, taste is actually mediated by what we call the taste buds. In other words, the sense organ for taste is known as the taste bud. The sense organ for taste is known as the taste bud. So the sense organ for taste are the taste buds. And if you would agree with me, the taste buds is actually ovoid in shape. It has an ovoid shape, and not only that, the taste buds are between 50 to 70 micron. They are between 50 to 70 micron. And usually, adults usually have 10,000 taste buds. Of course, this number is more in children. So children have more taste buds. And if you would agree with me, as one progresses in age, the number of taste buds begin to decline. It declines because as one advances in age, some of the taste buds begin to degenerate. And as they degenerate, the number begins to decline. So, let us talk about the situation or the location of taste buds. Now, it is important to know that taste buds are located more in the tongue. They are located in the papillae. The papillae of the tongue. So you find more taste bud. You find more taste bud in the papilla of the tongue. Also, you can find some taste buds in other areas in the oral cavity, such as the epiglottis. You can find taste buds in the epiglottis. You can find taste buds in the upper parts of the esophagus. So you can find taste buds in the upper esophagus. You can also find taste buds in the pharyngeal region. So you can find taste bud in the pharynx. So where can we find taste buds? We can, and also in the palate. You can find taste bud in the palate, particularly in the soft palate. You can find it around the soft palate. So where are taste buds situated or located? Taste buds are located more in the papillae of the tongue. They are located more in the papilla of the tongue. And also you can find taste buds in other regions of the oral cavity, such as the epiglottis, the upper part of the esophagus. You can find taste buds in the pharyngeal region, particularly in the oropharynx, around the oropharynx. Particularly around the oropharynx. And then you can also find taste buds in the palate, especially around the soft palate. So let's proceed. Let's talk about the taste buds that are located in the papilla of the tongue. Now, if you look at it, let me draw the diagram of the tongue. This is the typical diagram of the tongue. This is a typical diagram of the tongue. Now, the tongue is actually divided into two regions by a sulcus that is located posteriorly. So this sulcus is actually called, let me draw it here. This sulcus is called, the sulcus terminalis. Sulcus terminalis. So the sulcus terminalis divides the tongue into an anterior two-third and a posterior one-third. So this is the posterior one-third of the tongue, and this is the anterior two-third of the tongue. Now, if you would agree with me, we have about 4 types of papilla that are located in the tongue. The first one I will mention is known as fungiform papilla. Fungiform papilla. Fungiform papilla. The fungiform papilla is located in the tip of the tongue and in the anterior region of the tongue. So you can find it around this area. You can find the fungiform papilla around this area of the tongue. So all this area contains the fungiform papilla. So this is known as fungiform papilla. Now, the next papilla that you will see in the tongue is what is called the circumvallate papilla. The circumvallate papilla is actually located around the posterior part of the tongue. Now, in the posterior part of the tongue, you see this sulcus terminalis, right? The circumvallate papilla forms a V-shaped structure around this region. So this is where we have the circumvallate papilla. So this is circumvallate papilla. And then we have another type of papilla. That is located in the lateral border of the tongue. It is also located in the posterior part of the tongue. So, the lateral border, we have another type of papilla, which is also located in the posterior part of the tongue. So we call this one is called the foliate papilla. If you want to remember it, if you want to remember that it is located, excuse me, if you want to remember that it is located in the lateral border of the tongue, you look at the word foliate, lateral, right? Foliate papilla. It is located in the lateral part of the tongue and also in the posterior part of the tongue. And then there is another one which is called the filiform papilla. This one is actually located in the dorsal of the tongue, and it actually gives the tongue a friction-like nature. That is to say that, for instance, it is this papilla that allows you to be able to lick, ice cream. For those of you that like to lick ice cream, it is the filiform papilla that increases the friction of the tongue and enables you to lick your ice cream. So this one is actually called the filiform papilla. It is actually conical in shape. It has a conical shape, and it is located in the dorsal of the tongue. The fungiform papilla is round in shape. It is located at the tip of the tongue and also in the anterior two-thirds of the tongue. The circumvallate papilla is located in the posterior border of the tongue, and it forms a V-shaped structure around the posterior border of the tongue. Now let us zoom in on one of these papillas. Let's zoom in on one of these papilla and look at the structure. Of the papilla. So I think I can clean this now. Alright. So a typical taste bud looks like this. A typical taste bud usually has about 4 types of cells, and actually located in a typical taste bud. So let me mention the cells. The first one are the type 1 cell. The second one is type 2 cell. The third one is type 3 cell. The fourth one are the type 4 cell. So the type 1 cell is known as supporting cell. The function of type 2 cell is unknown. Type 3 cells are actually the taste cells. They are known as taste receptor cells. Or you can call it taste cells. Type 4 cells are known as basal cells. Now, the type 1 cells are supporting cells. Type 2 cells, the function of the type 2 cells are unknown. The type 3 cells are actually the taste cells or the taste receptors. And then type 4 cells are the basal cells. The basal cells are very important in the sense that they tend to, because the cells in the taste buds have a lifespan of 10 days. they have a lifespan of 10 days. So when they die, the basal cells are the ones that actually regenerate the taste cells. So they are very, very important. So let me draw the diagram of the board. Now, this is known as type 1 cells. This is type 1 cell, which is also known as supporting cell. Right? The type 1 cells are also called supporting cell. The next one is type 2 cell. So this one is type 2 cell, whose function is unknown. Then the next one are the type 3 cells. Now, like I mentioned earlier, the type 3 cells are the taste cells or the taste receptors. And then the last one are the basal cells. The basal cells. The basal cells, like I mentioned earlier on, these are the basal cells that are responsible for regenerating other cells. Now, the type 1, type 2, type 3 cells actually have cilia. They have cilia. And the cilia actually have microvilli. Right? Microvilli. Now, let me draw the cilia. These are the cilia. These are the cilia. These are the cilia. Now, these cilia actually open into, because this is actually modified epithelial cells. So, these cilia actually open into the mucosa of the oral cavity. So, and this area is actually known as the taste pore. Taste pore. So, the cilia actually go down to, they open into the taste pore, right? And they are responsible for picking chemicals, tasty chemicals, right? They are responsible for picking these chemicals like salts, sodium chloride, sugar, and the rest of them, right? Right? So that's the function of the cilia. The cilia. All right. So you can see we have the type 1 cells, the type 2 cells, the type 3 cells, and the basal cells. The type 1, type 2, type 3 cells all have cilia that opens into the what? The taste pore. And then we also have our nerve endings around this area. We also have our nerve endings around this area. These are the nerve endings that carries impulses to specific areas of the brain. All right. We normally have about 5 types of taste. We normally have 5 types of taste. We have, number 1 is the salty taste or salt taste. Number 2 is known as sour taste. Number 3 is known as bitter taste. Number 4, the first one is salty taste. The second one is sour taste. We have bitter taste, we have sweet taste. And the 5th one is known as umami. Umami. So this classification is based on the type of chemical, right? It is based on the type of chemical that is being sensed by the tongue, right? It is based by the type of chemical that is being sensed by the tongue. So based on the chemical that is sensed by the tongue, we have 5 types of taste, which are the salt taste, we have the sour taste, we have the bitter taste, we have the sweet taste, and then we have the umami. The umami is actually a Japanese word, and it is known as a very pleasant taste. Now, the salt taste is actually mediated by certain electrolytes like sodium, ion, right? It is mediated by certain electrolyte or ions such as sodium and even potassium ion, and in fact also chloride ion, right? So the salt taste is mediated by electrolytes or ions such as the sodium ion, the potassium ion, and the chloride ion. Now, depending on the concentration of these ions, right? When the concentration of these ions are high, then we tend to sense the salt taste. Now, the next one is called sour taste. Sour taste is mediated by protons. It is mediated by protons, such as the hydrogen ion, right? It is mediated by protons, such as hydrogen ion. The next one is the bitter taste. Bitter taste is actually mediated by organic substances like alkaloid and quinine, right? And then it is also important to know that the bitter taste has a very low threshold. It has a very low threshold, and then it has a survival value, right? It has a low threshold and it has a survival value. Because why I say that is because most of the poisons are actually bitter. So when you take in something that is bitter, you tend to refrain from it. So it has a low threshold and it has a very high survival value. Then the next one is sweet taste. Sweet taste is mediated by substances like sugar, right? Aldehyde, ketones, and even alcohol. So it is not only sugar that is sweet, right? Substances like organic substances like aldehyde, ketones, and alcohol are also sweet in nature. Then the umami is actually a pleasant taste, and like I mentioned earlier on, it is actually a Japanese word. It is a Japanese word, and it is mediated by substances such as what we call L-glutamate. L-glutamate. So let me run this again. Based on the chemical that is specific, we have 5 types of taste. The salt taste, which is mediated by ions or electrolytes such as sodium ion, potassium ion, and chloride ion. The next one is the sour taste, which is mediated by protons such as hydrogen ion. The third one is the bitter taste. The bitter taste is mediated by organic substances like alkaloid, quinine. And like I said, it has a low threshold and has a survival value. The next one is the sweet taste. The sweet taste is mediated by sugar, aldehyde, ketones, and alcohol. And then we have a very pleasant taste, which is umami, which is mediated by the L-glutamate. Now let us look at the sensing transduction mechanism of these various types of tastes. Right? Now, for the sensing mechanism, these two, the salt and sour tastes, are almost the same. Then these 3 are also the same. So we consider these 2 as one, and then consider this as one. Now let's go. The diagram is like this. Now, this is our taste receptor. These are taste receptor, right? These are taste receptor. And then, of course, we have some channels here. We have some channels, right? Now, these channels are known as epithelial sodium channel. This is epithelial sodium channel, right? Now, we also have another channel here. This one is calcium channel. That's calcium channel. This is epithelial sodium channel. This is epithelial sodium channel. Now, when you take in a salty food, right? And of course, salt contains sodium chloride. Now, when sodium chloride, let's say sodium ion, when sodium ion binds to this channel, right? When sodium ion binds to this channel, right? The channel is open and then it allows for the influx of sodium, right? It allows for the influx of sodium. Now, when sodium comes into the receptor, the cell, now what it does is that sodium goes down to activate this particular channel and allows for the influx of calcium ion, right? When calcium ion comes in, calcium ion binds to certain synaptic vesicles, right? They bind to certain synaptic vesicles, and they bind to certain synaptic vesicles. Now, these vesicles contain some neurotransmitters like serotonin, and acetylcholine, right? The neurotransmitters are serotonin and acetylcholine. So, and you know that in the cell, in the membrane of the cell, we have what we call synaptogens, right? Now, what calcium does is to mediate the interaction between these vesicles and the synaptogen, right? It mediates the interaction between the vesicles and the synaptogen. And then when that happens, the vesicle, the membrane of the vesicle fuses with the membrane of the cell, right? Of the taste cell, and then the neurotransmitter that is present in the vesicle will now be released, right? It will be released. Serotonin or ACH will be released. ACH, that is acetylcholine, will be released, right? And where will it be released? It will be released on the nerve fiber, right? It will be released on the nerve fiber that carries, which will carry taste impulse to specific areas of the brain. So let me take this again. Now, when sodium ion binds with the epithelial sodium channels, it opens the channels, allows sodium ion to flow into the taste cell. Now, when sodium ion flows into the taste cell, it binds to calcium ion and opens up this channel. Sorry, it binds to calcium channel and opens up this channel. When this channel is open, it allows for the influx of calcium. Calcium will now mediate the interaction between synaptogens and certain vesicles that are present within the taste cell. Now, when that happens, the membrane of the vesicle will bind with the membrane of the taste cell, and then when that happens, it will now dislodge the neurotransmitters that are located in the vesicle. I think we call that process exocytosis. We call it exocytosis. So, these particular neurotransmitters will now be released will be releasing, I think this is what we call synaptic clefts. Synaptic clefts. It will be released in the synaptic cleft, and then of course it will stimulate the nerve fiber, right? It will stimulate the nerve fiber that has the synaptic contact with the cell. And when that happens, it will result in the generation of action potential or impulse that will be transmitted along this nerve fiber to specific areas that are responsible for taste. Now, for the second one, which is sour taste. Sour taste is actually mediated. It is mediated by protons, such as hydrogen ion. So, if hydrogen ion binds with this channel, hydrogen ion, the channel will open and then there will be an influx of hydrogen ion. The hydrogen ion will now mediate, usually there's another channel here, which is potassium channel. What hydrogen ion does is to block this channel and prevent the efflux of potassium ion, right? It prevents, hydrogen ion blocks this channel and prevents the efflux of potassium ion. When this channel is blocked, and then when the efflux of potassium ion is prevented, it will result in the depolarization, right, of the taste cell or the taste receptor. Now, when that happens, the depolarization of these taste cells will now result in the opening of calcium channels, which will allow calcium to flow into the cell. When calcium flows into the cell, it will now mediate the interaction between the synaptogens and the synaptic vesicles. When that happens, the membrane or the synaptic vesicle will bind or fuse with the membrane of the taste receptor, and then exocytosis will occur. Serotonin and acetylcholine will be released, which will stimulate the nerve fiber that makes synaptic contact with the receptor, the taste receptor, and that will result in the generation of action potential, which will be transmitted along the nerve fiber to specific areas in the brain that are responsible for taste. So that is that for sour taste and also for salty taste. Now let us go to bitter taste, sweet taste, and umami. All of them are the same. I think I can train this. Now, for bitter taste, sweet taste, and umami taste, this one is actually mediated by a gene protein coupled receptor. It is mediated by a gene protein coupled receptor. So let me draw the diagram of this one. This one is actually mediated by a gene protein coupled receptor. Now, let's say this is our gene protein, right? And of course, this gene protein is known as transducin. Transducin. When an organic compound binds with this gene protein, right, it will activate this transducin, and transducin, when activated, will go and activate an enzyme that is present in the membrane of the taste receptor. And this enzyme is known as phospholipase C. Phospholipase C. Now, when phospholipase C is activated, it will result in it will act on what we call phosphorinositol bisphosphate, right? It will catalyze a reaction which will break down phosphorinositol bisphosphate to diacylglycerol, what we call DAG. DAG is diacylglycerol, and also inositol triphosphate. Now, the diacylglycerol will go down and activate what is known as protein kinase C. It will activate protein kinase C, and protein kinase C will go down to certain channels and phosphorylate this channel. So it will phosphorylate this particular channel, and when this channel is phosphorylated, it will allow for the influx of calcium ion. It will allow for the influx of calcium ion. Also, it is important to know that the inositol triphosphate has goes to the endoplasmic reticulum. It goes to certain channels. It binds with certain channels on the endoplasmic reticulum. Right? The endoplasmic reticulum contains lots of calcium ions. It contains lots of calcium ions. So when inositol triphosphate binds with this channel, it opens these channels, it opens up this channel and allows for the efflux of calcium ion from the endoplasmic reticulum. So this is smooth endoplasmic reticulum. Smooth endoplasmic. Okay, let me abbreviate it. Endoplasmic reticulum. Smooth endoplasmic reticulum. Right? Now, the inositol triphosphate will bind with this channel and will release the calcium. The calcium ion that are located in the smooth endoplasmic reticulum, and then calcium ion will be released. Right? When calcium ion is released, it goes down to the certain vesicles that contain serotonin and acetylcholine. Right? And then it mediates, just like we said earlier on, it mediates the interaction between these vesicles and certain synaptogens that are located in the receptor membrane. And when that happens, these vesicles will bind, the membrane of the vesicle will fuse with the membrane of the receptor, and when that happens, serotonin and acetylcholine will be released into the synaptic clefts, and then acetylcholine and serotonin, that's the neurotransmitters, will stimulate the nerve fibers that actually have synaptic contacts with the taste cell. And when that happens, it will result in the generation of action potential. It will result in the generation of action potential, which will be transmitted to specific areas of the brain that are implicated in taste. So you can see that we have been able to discuss the transduction mechanism of sour, bitter, sweet, and umami taste. We have discussed all of them. Right. So let's proceed to the next subtopic. Now, the next thing I will talk about is taste pathway. Taste pathway. Taste pathway. Now, when we talk about taste pathway, we are talking about the pathway that actually transmits the taste stimuli or impulses for taste. Now let me draw it down now. Right? This is the brain, this is cerebral cortex, this is the temporal lobe, and of course this is the thalamus, This one is known as the third ventricle. These are the ventricles of the brain. This is the lateral ventricle. This is the lateral ventricle. The lateral ventricle. Right? And then, let me draw Alright, this is the diagram of the brainstem. This is known as the gustatory area. This is the gustatory area. Gustatory area. This gustatory area consists of the nucleus of tractus solitarius. Nucleus of tractus solitarius. Right? It contains, the gustatory area is the nucleus of tractus solitarius that is actually located in the medulla. It is located in the medulla of the brain. Now, let me draw the diagram of the tongue. This is the tongue. Right, this is the tongue. And then let me draw the diagram of the pharynx. Alright. So this is the tongue, this is the tongue, and this is the pharynx. Now, when we talk about taste pathway, there are certain cranial nerves that are implicated in taste pathway. There are certain cranial nerves that are involved, that has an involvement in 7. Cranial nerve 7. We also have the glossopharyngeal nerve. The glossopharyngeal nerve, which is known as cranial nerve 9. And then we also have the vagus nerve. The vagus nerve is known as cranial nerve 10. So these are the nerves that are implicated or that has an involvement with the taste pathway. Now, the first one is actually a branch of the facial nerve, which is the corda tympanic branch of the facial nerve. Right? Now, the corda tympanic branch of the facial nerve actually arises from the anterior two-thirds of the tongue. So it arises, it supplies the anterior two-thirds of the tongue. It supplies the anterior two-thirds of the tongue and goes down, enters a ganglion. It enters a ganglion which is called the geniculate ganglion. We call it the geniculate ganglion. Right? And then goes and enters the medulla and synapses with the nucleus of tractus solitarius. Right? So the first order neuron is actually located in the geniculate ganglion. And the second order neuron is actually the nucleus of tractus solitarius. The second one is the glossopharyngeal nerve. The glossopharyngeal nerve arises from the posterior one-third. The posterior one-third of the tongue, and then goes down, enters the ganglion, This ganglion is actually called the petrosal ganglion, right? It is called the petrosal ganglion. Petrosal ganglion, right? So from here, from this ganglion, it enters the medulla and synapses with the nucleus of tractus solitarius. So the first order neuron is located in the petrosal ganglion, and the second order neuron is actually the nucleus of tractus solitarius. Now, the vagus nerve arises from other areas in the oral cavity, like the pharynx, the epiglottis, right, the palate, and the upper esophagus. So all these other regions actually are innervated by the vagus nerve. So these regions, all these regions are innervated by the vagus nerve, which enters a ganglion, which is known as, this ganglion is known as the nodose ganglion. It is known as the nodose ganglion. And then from here, it enters the medulla and synapses with the nucleus of tractus solitarius. So the first order neurons are located in this ganglion, in the geniculate ganglion, the petrosal ganglion, and the nodose ganglion. The second order neuron is located in the medulla, and of course is the nucleus of tractus solitarius. Now, the nucleus of tractus solitarius will give rise to certain fibers, right? It will give rise to certain fibers, and of course, these fibers form what we call the fibers run through what is referred to as this is known as the medial lemniscus. Medial lemniscus. Right? And it runs on the ipsilateral side. It runs on the ipsilateral side of the brain. That is to say that it runs on the same side of the brain. Right? It does not cross. The fiber does not cross. It doesn't go to the contralateral side, but it stays on the ipsilateral side and ascends upward down to the thalamus. So this structure is known as our thalamus. This is the thalamus. Now in the thalamus, it synapses with a certain nucleus of the thalamus, which is known as the VPN. VPN. Right? The VPN is known as the ventroposterolateral nucleus. Ventroposterolateral nucleus of the thalamus. Ventroposterolateral nucleus of the thalamus. Right? Now from here, fibers will now go down to an area in the parietal lobe, which is known as the postcentral gyrus. From that area, it will go down to the postcentral gyrus. From there, the fibers. You know, the postcentral gyrus is the area that is responsible for the conscious perception of taste. The postcentral gyrus is an area that is responsible for the conscious perception of taste. The conscious perception of taste. Now from there, fibers will now be tucked in, will go down to the temporal lobe and go to specific areas that are also implicated in taste. And these areas are what we call the anterior insula, and also another area that is called the frontal operculum. The frontal operculum. The frontal operculum. So let me take this again so that for better understanding, let me run through this again. Now, like I mentioned earlier on, I said that the taste pathway, it is simply the pathway that transmits taste sensation. It is the pathway that transmits taste sensation, right? And of course, it begins from the tongue and the pharynx. Now, in the tongue is divided into the anterior to third and the posterior to third. The anterior to third is supplied by the corda tympanic branch. The corda tympanic branch. This one is called the corda tympanic branch of the facial nerve. And of course, the facial nerve is cranial nerve 7. Cranial nerve 7. Right? Then, this nerve actually passes through, it enters the geniculate ganglion, where we have the first order neuron, and then goes down to the medulla and synapses with the nucleus of tractus solitarius. Now, the posterior to 1/3 actually is supplied by the glossopharyngeal nerve. The glossopharyngeal nerve is cranial nerve 9. From there, it goes down to the petrosal ganglion where we have the first order neuron, and then from there, it goes to the nucleus of tractus solitarius and synapses with the nucleus of tractus solitarius. The third one is the vagus nerve. This one is known as the glossopharyngeal nerve. This one is known as the vagus nerve. This is the vagus nerve. Now, the vagus nerve arises from other regions in the oral, like the pharynx. Right? The pharynx, the epiglottis, the palate, particularly the subpalate, right? And the upper part of the esophagus. And then it supplies this area and carries the information down to the locus ganglion. It carries fibers to the locus ganglion where we have the first order neuron. And then from there, the fibers enters the medulla and synapses with the nucleus of tractus solitarius. Now, the nucleus of tractus solitarius is the second order neuron. This neuron actually gives rise to fibers. It gives rise to fibers that runs through the medial meniscus, right? And goes down to the thalamus. In the thalamus, it synapses with the third order neuron, which is the VPN. The VPN is the ventroposteromedial nucleus of the thalamus. And then from there, the VPN gives rise to fibers that goes to the postcentral gyrus. The postcentral gyrus is an area that is responsible for the conscious perception of taste. And then from the fibers are redirected to an area that is known as an area in the temporal lobe that is known as the anterior insula and the frontal operculum. So this is the pathway. This is the pathway for taste, right? This is the pathway for taste. Now let us talk about. I think we are done with taste. We are done with taste. Now let's go to smell, olfaction. Let's talk about smell and olfaction. We are going to talk about smell and olfaction, and thereafter we are going to talk about vision, the visual pathway, and audition. We talk about the auditory pathway. Please excuse me. So let's talk about the physiology of olfaction. Physiology of olfaction. The physiology of olfaction. Olfaction is otherwise known as smell. Olfaction is otherwise known as smell. Now let me draw one or two diagrams to drive home my points. Smell is actually mediated by what we call olfactory receptors. And the olfactory receptors are located in the nasal cavity, right? They are located in the nasal cavity, particularly the nasal mucosa. The nasal mucosa is located in a region that is actually above the superior nasal concha, right? It is located above, quite above the superior nasal concha. Let me draw that on the board. Let me represent it on the board. Now this is the diagram of the cribriform plates. This is the cribriform plates, right? This is the cribriform plates of the ethmoid bone. This is cribriform plates. Of the ethmoid bone, right? Below the cribriform plates of the ethmoid bone, we have what is called the nasal mucosa. We have the nasal mucosa. Right? So this is the nasal mucosa. This is the cribriform plates of the ethmoid bone. Right? Now, in this cavity, we normally have what we call the olfactory receptors. The olfactory receptors actually has two processes. One is called the peripheral process, and another one is called the central process. So let me draw another one here. This is the peripheral process, and this is the central process. Let me draw another one here. This is the peripheral process, and this is the central process. Let me draw another one here. This is the peripheral process, this is the central process. Right? All right. So now, so this is called olfactory receptor. Olfactory receptor. The olfactory receptor has a peripheral process known as the olfactory rod. So this is known as olfactory rod. Olfactory receptor, olfactory rod. It ends in a knob-like structure that is known as olfactory knob. So this is olfactory knob. Right? Olfactory knob, olfactory knob, olfactory rod, olfactory receptors. The olfactory rod has some immotile cells that are immotile cilias, right? We call them olfactory cilias, or we call them olfactory hairs. They are immotile cilias. So this is olfactory hairs. Olfactory hairs. Right? The hairs actually are responsible, they have what we call odorant receptors. The hairs have odorant receptors and are responsible for picking certain odoriferous substances or molecules, right? That actually dissolve in the nasal mucosa. For instance, when there is a smell, for instance, if somebody farts, let me say someone's fat, let's say somebody farts, and of course fat contains methane, right? It contains methane. Now this methane is a gas that will go up, it will go up the nostril, and when it goes up the nostril, it goes to the mucosa, the olfactory mucosa. When it gets to the olfactory mucosa, it dissolves in the olfactory mucosa and he's picked by the odorant receptors that are located on the olfactory hairs. The odorant receptors located on the olfactory hairs actually pick up these odoriferous substances, and then when that happens, it will result in generation of receptor potential, which will result in generation of action potential that will be transmitted in the form of impulse down to the olfactory nerve. From the olfactory nerve, it will go to the olfactory tract, and then it goes to specific areas of the cortex. Now, like I said, I want to drive home my point. I want you to understand this thing very well. This is the olfactory receptor. This is olfactory receptor. This is olfactory rod. This is olfactory bulb. These are olfactory hairs. The olfactory hairs has what we call odorant receptors. Odorant receptors that picks odoriferous molecules or substances. Now, the olfactory hairs, okay, there are other cells. Apart from the olfactory receptors, there are other cells that are located in the nasal mucosa. Now, we have what we call the sustentacular cell. The sustentacular cell. These sustentacular cells are actually known as supporting cells, right? Sustentacular cells are known as supporting cells, and apart from the fact that they support the olfactory receptors, they are also responsible for producing mucus, right? They are also responsible for producing mucus. Now, we also have another one. We have a gland located in this region, which is known as the Bowman's gland. We have a gland that is known as the Bowman's gland. The Bowman's gland is responsible for secreting. This is called Bowman's gland. Bowman's gland. This one is called sustentacular cell. Sustentacular cells. Now, the Bowman's gland is responsible for secreting mucus. The Bowman's gland is responsible for secreting mucus. The sustentacular cells are supporting cells, they support the receptor cells, and are also responsible for secreting mucus. Then we have another one. We have another cell. This one is actually called basal cell, right? It is called basal cell. We have it around this area. These are the basal cells. Basal cells. They are responsible for reproducing or regenerating damaged olfactory receptors. When the olfactory receptors are damaged, the basal cells are responsible for reproducing or regenerating more olfactory receptors. Now, like I said, the olfactory receptors has two processes. One is the peripheral process, and the other one is the central process. The central process actually are zones of these particular receptor cells which come together to form what is known as the olfactory nerve. It forms the olfactory nerve. So this is known as olfactory nerve. This is olfactory nerve. Olfactory nerve. The olfactory nerve is actually formed by olfactory receptors, right? It is formed by their zones, the central processes of the olfactory receptors. They actually come together to form the olfactory nerves. And where do they go to? They go to a structure that is known as the olfactory bulb. The olfactory bulb lies around the under surface of the frontal lobe, right? So this is where we have the olfactory bulb. This is olfactory bulb. This is olfactory bulb. This is olfactory bulb. This is olfactory bulb. Now, the olfactory bulb has some cells, right? The first one I'll mention is actually known as the mitral cell. The mitral cell, right? The mitral cells gives rise to dendrites that synapses with the axons of the olfactory nerve, right? And they form a structure that is known as the olfactory glomeruli. So this is known as olfactory glomeruli. Olfactory glomeruli. It is different from the glomeruli of the kidney. So let me draw another one here. Right? This is olfactory glomeruli. Right? Then we have another one that is called the tufted cells. We have another one that is called the tufted cells. The tufted cell is also located in the olfactory bulb. The tufted cell is also located in the olfactory bulb. So this one is known as olfactory. This is known as mitral cell. Mitral cell. This one is known as tufted. This is known as tufted cell. Tufted cell. Right? We have the mitral cell, and then we have the tufted cell. Then we also have other cells. Right? We also have other cells. Now, the difference between mitral cells, there's a difference between mitral cells and tufted cells. Mitral cells are larger, right? They are larger. And they usually have one synaptic contact with olfactory nerve, right? They normally have one synaptic contact just like this. They normally have one synaptic contact, right, with olfactory nerve. But the tufted cells are actually large, smaller in nature. They are smaller in nature and they have multiple synaptic contacts. Now let's say this is a tufted cell, right? It will have multiple synaptic contacts with olfactory nerve, right? So this one is known as mitral cell. Mitral cell. This is known as tufted cell. Tufted cell. The tufted cells are smaller than the mitral cell, but they have multiple synaptic contacts with the olfactory nerve. Whereas the mitral cells usually have a single synaptic contact with the olfactory nerve. The olfactory nerve. Now, we have other cells like the granule cells. The granule cells, right? The granule cells do not have axons. The granule cells do not have axons, but they usually have synaptic contacts with they have synaptic contacts with the lateral fibers, right? They have synaptic contacts with the lateral fibers of the mitral cells and the tufted cells, right? Now, what happens is that they are actually inhibitory in nature. When an action potential reaches the mitral cell, right, the mitral cell will be activated and then it will release glutamate, right? The glutamate will stimulate the granule cells and the granule cells will release GABA. Now, what does GABA do? GABA is responsible for inhibiting the mitral cells. So what the granule cell does is that it actually inhibits certain mitral cells and allows certain cells to actually fire, right? It inhibits certain cells that has low excitatory postsynaptic potential and allows the mitral cell that has high excitatory postsynaptic potential to actually fire and transmit impulses. So that's the job of the granule cell. So this is known as granule cell. Now the next one is what we call periglomerular cell. We have periglomerular cell, which actually it has synaptic contact with two. It usually has synaptic contact with two glomeruli, right? It usually has synaptic contact with two or more glomeruli. So we call this periglomerular cell, right? And then we also have what we call the anterior olfactory nucleus. We have what we call the anterior olfactory nucleus. Now this nucleus actually transmits impulses. It sends out fibers that goes to activate the granule cells, right? It stimulates the granule cells to actually release GABA and inhibit certain mitral cells. All right. Now, it is important to know that the mitral cells and the ductal cells gives rise to azones, right? And these azones actually extend from the olfactory bulb and go down to the olfactory cortex. They go down to certain areas of the olfactory cortex, right? Now, these particular azones actually form what is known as the olfactory tracts. Olfactory tracts. The azones of the mitral cells and the ductal cells form what is referred to as the olfactory tract. So this is the olfactory tract, this is the olfactory bulb, these are olfactory receptors, this is known as olfactory nerve, this is glomeruli and all that. Right. Now, let us talk about the cause of these olfactory tracts. Let's see how it enters certain portions of the cortex and excites those areas. Now, let me draw the diagram of the brain. Right? This is the optic chiasma. Right? This is the brainstem. This is the midbrain. This is the midbrain. Right? This is the frontal lobe. This is the frontal lobe. This is the temporal lobe. Now, if you would agree with me, we normally have certain olfactory receptors, right? We have certain olfactory receptors that goes down, right? That goes down to the olfactory bulb, right? We have certain olfactory receptors that goes down to the olfactory bulb, right? Now, in the olfactory bulb, these olfactory receptors synapses with the mitral cells and the ductal cells, right? And then they give rise to azones that forms what we call the olfactory tracts, right? They form what is called the olfactory tracts. Now, when the olfactory tract gets to an area that is known as the lateral olfactory striae, it gives rise to the lateral olfactory striae and also gives rise to the medial olfactory striae. Now, the medial olfactory striae crosses the midline, right? The midline is around the region of the anterior commission. This is the anterior commission. Anterior commission. Right? This area is known as the area. This area is the anterior perforated substance. That's the anterior perforated substance. Now around that area, the olfactory tract divides into two, forming the lateral olfactory striae and the medial olfactory striae. So the medial olfactory striae crosses the midline around the anterior commissure and goes to the opposite olfactory bulb, right? And then, so this is medial olfactory striae, right? This is lateral olfactory striae. Now the lateral olfactory striae now goes down to the cortex, to certain areas of the cortex, and these areas are, we have the olfactory tubercle, olfactory, the piriform area, or the piriform area, right? And then we have an area that is known as the entorhinal cortex. Entorhinal cortex. This cortex, the entorhinal cortex is a component of what is referred to as the parahippocampal gyrus, right? It is a component of the parahippocampal gyrus, and it is known as area 28. It is known as area 28 in the Broadman's classification. And this entorhinal cortex is actually known as the secondary, it is the secondary olfactory cortex. It is known as the secondary olfactory cortex. Whereas the periamygdala area and the piriform area are known as the primary olfactory cortex. Right? Now these areas can also give out fibers that passes through the thalamus, right? Through the thalamus, and from the thalamus they go down to areas such as the orbitofrontal cortex, the orbitofrontal gyrus, and the subcallosal gyrus, the subcallosal gyrus, and the prefrontal cortex, right? So these areas are responsible for the conscious perception of taste. They are responsible for the conscious perception of, of smell. They are responsible for the conscious perception of smell. So it is important to know that from the olfactory nerve, from the olfactory receptor, right? The olfactory receptor will be right to the olfactory nerve, which will go into the olfactory bulb, right? And synapse with the mitral and the tufted cells, and then as zones of the mitral and tufted cells will actually go down. Of course, the mitral cells are found in the olfactory bulb. They will go down to the anterior perforated substance, right? And divide into two, right? They will divide into two, giving rise to the medial olfactory striae and the lateral olfactory striae, right? Now the medial olfactory striae will cross the midline, right? The midline is around the anterior commissure, and then it will go to the opposite olfactory bulb. Then the lateral olfactory striae will go down to the olfactory cortex, and the olfactory cortex contains the certain regions such as the olfactory tubercle, the anterior olfactory nucleus, the periamygdala area, the piriform area, and the entorhinal cortex. These areas are areas that are located in the limbic system, right? They are limbic areas. And of course, like I mentioned earlier, we have the primary olfactory cortex, which consists of the periamygdala area and the piriform area. And then we have the secondary olfactory area, which is known as the entorhinal cortex. The entorhinal cortex is known as area 28. Area 28 is also known and the prefrontal cortex, and these areas are responsible for the conscious perception of smell. They are responsible for the conscious perception of smell. Now let us look at the sensory transduction mechanism that takes place in the olfactory receptors. Let's go to that before we call it a day. Now, if you would agree with me, we have several olfactory receptors. Now, these olfactory receptors actually have their specific odorant receptors, right? They have their specific odorant receptors, right? These are olfactory, all these are olfactory receptors. Now each olfactory receptor has a unique odorant receptor, an odorant receptor that is specific to that particular receptors. And of course, if you agree with me, we have several odorant molecules that are found in our environment, lots of them, right? So let's say this is a particular odor. Let's say this is a particular odor. Let's also say that this one is also a particular odor. This is a particular odor, right? And this is also a particular odor, right? Now, if you agree with me, this particular odor can actually bind with this particular odorant receptor and can maybe also bind with this odorant receptor and can bind with these odorant receptors, right? One odorant receptor can bind to several odorants. One odorant molecule can bind to several odorant receptors, right? But one odorant receptor is specific to that particular olfactory, to one particular olfactory receptor. So this other odorant receptor can bind with this and can bind with this. This other one can bind with this, can bind with this, and can also bind with this. This one may bind with this alone, and this one can bind with this, can bind with this, and may have a tendency to bind with this, right? So it is important to know that each olfactory receptor has a specific odorant receptor that is unique or specific to that olfactory receptor. But then, like I said, we have several odorant molecules that have a tendency to bind with more than one olfactory receptor, right? Now, let us talk about the sensory transduction mechanism that occurs in the olfactory receptor. Now, if you will agree with me, let me draw okay. Let's say this is our receptor, and these receptors are actually G protein coupled receptors. So they are G protein coupled receptors, right? Now, when a particular receptor, right? It stimulates this receptor. When this receptor is stimulated, it results in the displacement of GDP. When GDP is displaced, it results, it's when GDP is displaced, GTP becomes attached to the receptor. And when GTP is attached to the receptor, it activates this receptor. When the receptor is activated, of course, this receptor is actually a trimeric receptor that has an alpha, beta, and gamma components. Now, when this receptor is activated, the alpha component will be activated and will be displaced, right? It will be dissociated from the beta and gamma dimeric components. Now, the alpha component will activate will go to the membrane and stimulate and activate an enzyme in the membrane. This enzyme is known as adenyl cyclase. Adenyl cyclase. If you don't call it adenyl cyclase, you call it adenyl cyclase. Now, when adenyl cyclase is activated, it catalyzes a reaction which involves the conversion of ATP to cyclic AMP. Cyclic AMP, right? When cyclic AMP is formed, it goes down to certain channels, ion channels, activates the ion channels, right? And allows for the influx of sodium ion. It allows for the influx of sodium ion, right? Now, when sodium ion comes into the cell, it changes the membrane potential of the cell. When it changes the membrane potential of the cell will now become, of course, it will become electropositive, right? The electropositivity it will become electropositive, which will change the membrane potential and results in the development of what is called receptor potential. Receptor potential. Now, the receptor potential is a graded potential, right? The receptor potential is a graded potential. Now, when the frequency of this potential increases, right? When the intensity of the molecule increases, the frequency of this potential also increases, which will result in the increase in the threshold of the potential. When the threshold of the potential is increased, it will result in generation of action potential. Now, let me draw another diagram, which I believe you are familiar with. Now, look at this diagram, right? Now, this is our membrane potential, right? Now, this is known as the threshold potential. This is threshold potential, right? This is our membrane potential, our resting membrane potential, right? Now, when sodium ion comes into the cell and changes the membrane potential of this cell, what will happen is that the membrane potential will change, and then when it changes, it results in the development of what is called receptor potential. Receptor potential, right? Now, when the receptor potential reaches a certain threshold, let's say it reaches minus 55 millivolts, right? When it gets to minus 55 millivolts, right, it spikes and results in the generation of what is known as action potential. The action potential, right, is what is actually carried by olfactory nerve down to specific areas of the olfactory cortex. And of course, when that happens, then there will now be a conscious perception of smell. There will be a conscious perception of smell. So, I think we have come to the end of this class. We have come to the end of this class. In our next class, we are going to talk about that is next Saturday, we are going to talk about the visual pathway. We are going to talk about vision, right? Of course, we'll talk about the visual pathway, right? And then, thereafter, subsequently, we'll talk about audition, that is hearing. And then we'll talk about the auditory pathway. We'll talk about the auditory pathway. Then, in our physical classes, we'll discuss other aspects of neurophysiology. I think we have talked about the somatic, the somatosensory system. We've talked about the dorsal columnar medial system. When next we meet in class, we are going to talk about the trigeminal pathway, right? We are going to talk about the trigeminal pathway because it carries sensation from the face, right, to specific areas of the brain. And then, when we are done with that, we will now talk about the anterolateral system, right? When we are done with the anterolateral system, then we are going to talk about we are going to talk about other areas of neurophysiology. Well, anyway, I want to thank you for coming to this class, for attending this class. Thank you for your time. Thank you for your patience. And I hope to see you in our next class on Saturday, and I also hope to see you in our next class on Wednesday. On Wednesday, we'll be having a class in the university. Thank you very much. Thank you. I hope to see you in our next class. Well done. Have a nice day.